Good morning or good afternoon, everybody. Thank you for giving me the opportunity to share a couple of my studies controlling botrytis and other fruit rots in strawberry. I'm going to share those details with you now. Um, First, I want to bring your attention to this slide where I put together a list of biopesticides and their categories for controlling arthropods, weeds, nematodes, mollusks, and diseases. On the top right corner, you see in the blue various biopesticides based on bacteria, fungi, viruses, botanicals, and toxic metabolites or other categories that are available for controlling a wide variety of diseases, including botrytis and other fruit rots. This is just to give you an idea of what kind of active ingredients are available and that can be used for controlling one or more diseases. Then botrytis fruit rot or gray mold is an important fruit and foliar disease in strawberry, and it is also a disease in many other crops. And what you are seeing here is a strawberry that is showing botrytis infection and spore production. It is caused by Botrytis cinerea, and what happens is this fungus can survive in the plant debris and also infect flowers and stays as a systemic in plant tissue. So although you see the infection primarily in fruits, you can have the infection in the plant tissue. And when the infection is severe, it could even destroy the flowers. So one of the strategies that some growers successfully use is to apply uh, fungicides for controlling this disease in the flowers as well, not just on the fruits. So this slide shows different stages of development of this disease and how it can progress. And these millions of spores on these fruits can serve as a source of a further infection in the field. So, irrigation or a splash of rain or cultural practices that disturb the fruit or plants could be distributing the inoculum. So we need to be aware of this potential dispersal within the field if there is any fruit left and in the field that is carrying these spores. So sanitation is one of the things that could be done when a fruit with this infection or spores is detected in the field. So now I also want to mention a couple of other diseases that we all commonly see in strawberry, and these were included because we had co-infections of these rhizopus fruit rot and mucor fruit rot in during the studies. So some of the data might include all of them because it is difficult to uh, distinguish unless there is a clear indication that it is a gray mold only. Sometimes there is a co-infection of others growing on the fruit during the observation. So the data also includes at some points the ratings for other diseases. So now the first I want to show the 2019 study. It was conducted at a research station in a small plot trial with the replications, 4 replications. And this is the experimental design I had. We had several treatments and they were randomized within each block, as you see on the right side. And then applied the fungicides with this backpack carbon dioxide pressurized backpack sprayer, and then applied on 4 dates and then harvested 3 days after each spray. But we missed one of them because of we had a rain event. And what we typically do for a study like this is to harvest and put these strawberries in plastic containers at the room temperature and observe the fungal growth 3 and 5 days after harvest. So this is the field where I conducted both these studies, and this is actually from 1 year, and we have these. What you're seeing is on the left side. This is the study plot for botrytis control, and these are the small plots, and being sprayed. And these are the treatments, 5 categories of treatments, actually, not the exact treatments. These are 5 categories of treatments I had during that study. The first, the one on the right, the column on the right side shows what those were. The first 4 materials were chemical fungicides that belong to different mode of action groups. And then I had this fatty acid based product. Then I had a couple of products that are based on eugenol, geraniol, and thymol. It was a number product at that time, and I just happened to look up the information online about what that active ingredient was. And the second one, what you see here is this 650004 is also based on that, but I couldn't find any information based on what they told me at the time. This is also similar to the other product. So I presume it has probably different proportions of the same essential oil active ingredients. And the next one is this Pseudomonas chlororaphis, a strain of that. It's a bacterial product. And the last two are Beauveria bassiana and Metarhizium anisopliae isolates. These are entomopathogenic fungi, and these are unformulated. These are the isolates I have for several years, probably 60 years or so. And they were initially meant for arthropod control, and I started to see some impact of these fungi in not necessarily these isolates, but other isolates of these fungi have been in the recent years studied for their roles other than killing arthropods. So we noticed that they promote plant growth, they endophytically colonize plants and offer protection against herbivores, and they also antagonize plant pathogens. So I studied these two for controlling these two fungi for controlling fusarium in cotton, and the same isolates for against Macrophomina fasciolina, which causes charcoal rot in strawberry. So because of that, I included them as an in the study to see if the growers are applying these fungi, the commercial formulations of these fungi for controlling arthropods, they might be getting additional benefit in terms of disease control as well. So this contributes to reducing the fungicide applications. So the main point here is that there were 5 categories of fungicides, and those were used in these 13 treatments. So the first one was just a rotation of different chemical fungicides as a standard. That is, treatment number 2. And then 3, 4, and 5 were different, these botanical extracts. And then the 6 and 7 were botanicals alternated with this microbial material, the bacterial fungicide. Then the 8 and 9 were chemicals alternated with botanicals. And then 10 and 11 were different concentrations of these organic oils, the fatty acids. And then 12 and 13 were mirivan and pristine alternated with these 2 entomopathogenic fungi. So this is what I have for disease rating. So we harvest them and put them in these clamshells and look at the disease 3 days and 5 days after harvest. And depending on the percentage of coverage, extent of fungal growth on these fruits, we would rate them accordingly, either 1, 2, 3, or 4. Here you can see on the top box on the left, this would be 1, rated 1, because infection was less than 25%. For all those berries in that clamshell. Then these were the disease ratings before we applied these treatments. And there was some variation among these, as you can see, but they were not significantly different. And then these were the data or the disease ratings after each of those applications. The first one and second application and third one we lost because of the rain. And then again, after the fourth spray application. You can see there were differences among the level of infection among these different treatments here. We consistently had higher infection from this untreated control, number one. And then number two was that chemical treatment. I hope you can see the cursor I'm showing here. And then here we had a significant control here too. And then again, after the fourth application, but rest of them varied, which is fine depending on the active ingredient that was used and depending on the application rate and how they were alternated with other ones. So we do see the important point is here is that we didn't have very high infection levels. As you can see, the highest we saw was slightly over 0.5 rating after the fourth application, but rest of them were around that 0.5. So that means like less than 25%. So if you combine the ratings for all those three harvest days, so this is what we see. Significantly higher in untreated control compared to many other treatments. And then we had a very good control from chemical fungicide applications. And what I want to bring your attention was other ones. It is not just that, the statistically significant difference among some of these treatments here, but these one rate of these fatty acids here, and then some of these botanicals, and also these chemicals alternated with the Beauveria bassiana. All of them provided significant control, not necessarily as good as chemical fungicides, but they did provide some significant control of botrytis. And as you already know, botrytis was one of those pathogens that develops resistance to fungicide treatments, and it is a common fungus. As a result of that, frequent fungicide applications are also very common. And when we are relying on one kind of a fungicide or similar ones, then if fungus or any pest typically develops resistance very quickly, and botrytis resistance is very well known. So it is important for us to look at other alternatives, non-chemical alternatives. And then when you look at these non-chemicals, again, there are a wide variety. As I showed in the first slide, we have bacteria and botanical extracts and fungi and so on. So it is always a good idea to look at how effective they are and then try to incorporate them into an IPM program. So these were the data from 2019 study. And then for the 2020 study, the treatments were slightly different. I had this Elevate only as the chemical standard, then Surefel, which is Bacillus faciens, and then Problad verde. It is from sweet lupin seeds. Then it is a polypeptide obtained from these seeds, and it was applied along with this cinerate, cinnamon oil-based fungicide at different rates. And then this one was also Problad verde with cinerate, but at but Problad applied at a lower rate compared to the other treatment. So these were the treatments. We just had a few ones in this study, and this is what we did. For the first study, we started applying and then, took the ratings. But here we removed the flowers and fruits before the first application. So we wanted to see if there was nothing that is actually, trying to spread. Do we have something that is systemic or something that comes after we start spraying, and what is the impact of these treatments and the disease development under those circumstances? So treatments were applied again on these different dates, and then observations were taken after 3 or 4 days after, and then we did the same thing for disease rating, 3 and 5 days after each harvest. So this is what I found from the study. These were 4 observation dates. And the important thing is that important but unfortunate that there were no statistically significant differences among these treatments. So the variation was higher, and we didn't see any differences. But all that you see is these numerical differences. We had, untreated control had higher disease here, here too, but, this treatment had more than that. Because there were no differences, I am not going to make any statements that one worked better than the other, but these studies are important for us to, at least to gather preliminary data and see how they're working under the field conditions. So if you combine the data for all these 4 observation dates, this is what you see. Elevate the fungicide, chemical fungicide seem to have lesser infection rate compared to untreated control. And Serifel, the Bacillus aeruginosa also had more or less the same level. This is what you see. There seems to be some kind of impact of these treatments, and obviously we need to do more studies to have solid data and significant differences. So the conclusions from both these studies, but mainly from the 2019 study, was that rotating various chemical fungicides worked very well. It provided a fair suppression of the disease, but at the same time, non-chemical alternatives also worked, but their efficacy varied depending on what was used, and the rate and the active ingredient and the rotation among them. But as I mentioned earlier, it is important to note that we always have to look at multiple control strategies, not just spraying fungicides, but maintaining good soil health and plant health. There are several studies showing that biostimulant materials trigger plant defenses and offer protection. So we should try to take advantage of those for maintaining general health of the plant. And we should also remember that when we are applying those other materials, such as entomopathogenic fungi, they might be providing control, some sort of control against these plant pathogens too, so we can reduce the fungicide application. So having a good understanding of what these cultural practices do, sanitation and applying are using these biostimulants and chemical and biological fungicides and so on, it helps us to develop a strategy that provides effective control and a strategy that is ideal for that particular crop and situation. So this is where we are with these studies, and I would like to thank my technicians Hamza and Marjan for providing assistance to collect the data and take all these ratings. And I would also like to thank these companies for providing financial assistance for these studies. And then I quickly want to mention that there were several YouTube presentations that I have been posting recently on different topics using biologicals and IPM and so on. So if you find those topics interesting, you might want to take a look at them when you get a chance. And with this, I end my talk, and you can look at my publications based on these studies and other studies at these electronic journals I have, and you can also feel free to communicate with me when needed. And if there are any questions, I would be glad to answer. And once again, thank you for giving me the opportunity to share this information with you.